So I'm Catriona Jamieson. I go by Cat. As usual, Luigi gave a spectacular talk. Thank you, Luigi. A lot of us owe our careers to Luigi, as Ellison started with. I just came off study section this morning, so it reminds me of how important it is to have this community pulled together for this particular meeting, because a lot of things that we think about the truth when it comes to stem cells that are self-evident aren't to people on NIH study section. So I'm particularly grateful to CIRM, the California Institute for Regenerative Medicine. I saw Maria Milan when I was running in here earlier. Thank you to you and your team for allowing us to do this. We know that you have to go through the 3 phases of the truth with any new discovery. First, it's ridiculed, then it's vehemently opposed, and then it's accepted as self-evident. So it turns out, at least by my experience on study section, it's not self-evident yet, but we're getting there. And it's specifically when it comes to translating to the clinic. I think Luigi's done a phenomenal job of making it look easy. We know it's really hard. And so in our Alpha Clinic, we're trying to do that. What I'd like to talk about today is targeted innate immune response pathways in cancer stem cells. I'll be the first speaker. We each get about 15 minutes, and then we'll have a panel discussion at the end, so we'll keep the questions to the end. I'll talk about antiviral immune responses in cancer stem cells. Then Jeremy Rich will regale us with his advances in glioblastoma stem cell targeting, which is very unique and interesting. Then Christine Brown will talk about her amazing work with CAR T-cell targeting of GBM stem cells as well, and just got a very nice CIRM grant for that. And then Travis Young will talk about switchable CAR T-cells from Caliber. Anyway, to begin, why do innate immune responses matter? And what about our blood? What about when our blood goes bad? So I just wrote this Nature News and Views, and I called it étude brute. And they said, nobody will understand what that means. And I said, didn't anybody else have to read Julius Caesar when they were in high school? Because I did. And the idea is your best friend turns against you, and that's what can happen to our blood. So this is called clonal hematopoiesis. When our blood stem cells decide to mutate themselves in response to toxins and environmental agents, including viruses, what happens? Well, it turns out these give rise to pre-leukemic stem cells, and what was recently published in a very nice paper in Cell Stem Cell by Ross Lavina and colleagues is those very mutated hematopoietic stem cells can promote solid tumor progression. So they provide the right instructive cues to the solid tumors the ticket to just take off, and it can also give rise to acute myeloid leukemia. So basically it's a double whammy. You get 2 hits with one, and they're not good hits. So then the idea is, how do we understand how these leukemia stem cells evolve, and what is it in particular that we may be able to dial down in terms of activity? Well, it turns out it's the innate sensors of exogenous RNA and DNA that are working against us when they're turned on at too high a level for too long. And these are the ADARs and the APOBECs in our system. So these are like our endogenous CRISPRs. So ADAR, adenosine deaminase associated with RNA1, edits RNA that's cytosolic and thought to be exogenous. And then APOBECs edit DNA primarily, and they can introduce mutations. So we think usually that the innate immune response, those cells that are the first line of defense, should be our friends, and they instruct other cells, the adaptive component of the immune system, to then really go in and start attacking. Those are the T and B cells. But if the innate immune response through ADARs and APOBECs are turned on for too long, they can create a problem. So ADAR, I'll start with this. These are enzymes that evolve to protect our stem cells from retroviral integration. And they're very important. They prevent stem cells from being corrupted by these viral sequences. They're so important that we have 3 of them in humans, ADAR1, 2, and 3. With ADAR1, that's a primary RNA editase in humans, and there are 2 versions of that. ADARP110 is the nuclear component. ADARP150 is the interferon gamma-inducible form that's a primary antiviral editase. And what these ADARs do is they edit adenosine to inosine, which is subsequently read as aguanosine. So what do they do? Well, ADARs are really important for determining transcriptomic diversity in humans. They can rewrite, recode, and rewire the human transcriptome, and they work primarily in the context of primate-specific ALU sequences. So ALU sequences, when I was in grad school, were thought to be junk. People said, these are junk, why do you care? Except for one person who was in our Department of Microbiology at the University of British Columbia, and that was Mike Smith. He said, I think allo sequences matter. And at the time, we didn't know there were 10% of our genome, but now, of course, we know. And it turns out that at least one of the functions of allo sequences is to form secondary structure that these ADARs can go in and bind to, and then edit the RNA within that double-stranded RNA structure. Mike Smith, as you probably remember, won the Nobel Prize for site-directed mutagenesis, but his main interest before he won the Nobel Prize was to work on these allo sequences. Anyway, so what ADARs do is they can edit these exonized allos, and they can also edit microRNAs because they have secondary structure, 3' untranslated regions, and long non-coding RNAs. And again, they're really part of our antiviral response, and they're triggered by interferon gamma. So what do they do to RNA? We know that there are epigenetic modifications to DNA, but what about RNA? So the field of the epitranscriptome has evolved, so we know that RNA can be modified post-transcriptionally. It can be methylated. N6-methyladenosine actually prevents adenosine editing. And then we know that ADAR can edit these transcripts and then change the outcome. So basically, we've been particularly interested in seeing what ADAR does in terms of allowing dormant cancer stem cells to basically clone themselves. These dormant cancer stem cells evade therapies that target dividing cells because they're asleep. And so how do you target those? Well, we worked with Roger Chen's postdoc at Tsushi Miyawaki to really change the FUCI system, which is standing for fluorescence ubiquitination cell cycle indicator, so that it was a single lentiviral vector. Gabriel Pineda did this in the lab together with Katie Lennon. And basically what it tells you is if a cell is in G0, G1, it's red. If it's SG2, it's green. And if it's dividing, it's yellow. So basically, this allows us to track populations of cells on very specific microenvironments. Sorry, this is a fancy system here. These are 293 cells. These are normal cells. And these are the cancer stem cell population. So you can see that in chronic myeloid leukemia, a lot of the cells are quiescent. They don't really want to wake up, and they're more adherent to the microenvironment. So that's a problem because we can't target these cells with a standard therapy for chronic myeloid leukemia, which is Gleevec. So if people stop Gleevec, 60% of people relapse within 6 months, and then the next patients relapse much faster. And those are the people that had no detectable BCR-ABL transcripts. So what do we do about that? Well, we really have to find the drivers of progression of chronic myeloid leukemia and other malignancies. And so by understanding what fuels activation of ADAR, I think we'll be able to do that. We know in CML blast crisis, that's when they switch from the earlier phase to the untreatable phase, they really upregulate interferon gamma. And this leads to activation of ADAR. So they activate that ADAR P150, which is the red line here, and that we know is going to cause some problems. What we found and published last year in Cell Stem Cell is that edits the differentiation-inducing microRNA family, the LET7 family. So that LET7 family is edited at the DROSHA cleavage site and also the dicer cleavage site. And if we introduce that edit, we can actually prevent maturation of the LET7 microRNA, and then also that leads to increased expression of some of the Wnt targets that normally would be targeted by LET7. So what that does is it enhances leukemia stem cell cloning potential, and we know we can inhibit that with a selective JAK2 inhibitor because that prevents STAT activation of ADAR. So that's the good news. We have a JAK2 inhibitor. We can repurpose that and use it to at least dial down ADAR activity. We know that this is important not just for leukemia, but for other cancers. Han Liang and Gordon Mills published that in 17 different tumor types, ADAR-mediated ATI editing is activated, and that turns out to be really important for tumor progression and also for therapeutic resistance. And then finally, in myeloma, we found that ADAR is activated because of 1Q amplification. ADAR is at 1Q21, right next to IL-6 receptor. This occurs in about a third of patients. These patients have a worse prognosis, and you can see those with a higher ADAR level have a poorer survival. This occurs because ADAR edits the self-renewal agonist GLI. And so GLI hangs around longer because it can no longer be degraded by its natural inhibitor, SUFU. We see GLI editing fairly easily, and then that is absolutely vital for allowing myeloma regeneration. And we see this in vivo. So we made a myeloma model. This is Leslie Cruz and Elisa Lazzari, who was very well trained in Milan. I don't think you trained her, Luigi, but she's well trained nonetheless. Anyway, she made a humanized mouse model of myeloma. And what we see is we get the, I'll just use the cursor here. We see the bioluminescent engraftment of myeloma. You can pick up the myeloma light chain and engraftment by FACS. When we see engraftment, they also form solid tumor collections of myeloma. Those are called plasmacytomas. And basically, we can inhibit myeloma regeneration by ADAR knockdown in the serial transplantation setting. So this paper is just coming out to summarize the myeloma piece. Basically, what you get in about a third of patients with myeloma is 1Q amplification. They amplify ADAR and IL-6. IL-6 receptor allows them to be more sensitive to exogenous IL-6, and they really activate ADAR. APOBECs, as I mentioned, can also be activated. These are deaminases that edit C to T. They're also involved in malignant progression. And what we found recently, just as the last slide, this is it, ADAR edits APOBEC. So the RNA editor is editing the DNA editor. So that means that extrinsic cues that we get for the microenvironment can actually introduce DNA mutations and activate these APOBECs 3D and 3G. So in essence, if we want to stop this mutagenesis process that goes on as we age, we really have to block the unbridled activity of these antiviral enzymes, ADAR and APOBEC. And I think we'll be able to get there, but it takes a village. So I'd really like to thank our team, not just our lab, but our Division of Regenerative Medicine, and also our Alpha Clinic and all of our colleagues here in San Diego, and I'd like to thank SERM. So that's it for me. I know that was a quick whirlwind tour, but I'd be happy to take questions at the end. Jeremy Rich is our next speaker. So